Now I would like to invite our next presenter, Naeem Ahmad from Nirji. Naeem Ahmad from Nirji.
Good afternoon, everybody. My name is Naeem Ahmad, and I am a PhD candidate from UT. The topic I am going to present today is genetic analysis of neurological disorders in consanguineous Pakistani families and their implications for routine genetic testing in healthcare practices. This work was conducted in Human Molecular Genetics Lab of NIMG. First of all, for a little bit of background, earlier today, one of our guest speakers was talking about the disease burden of neurological disorders. I would like to build up on that. Neurological disorders are not only abundant globally, but they are also the leading cause of disabilities worldwide, and they are the second largest cause of mortality worldwide. And according to WHO data, the death rate is more in low and middle income countries like Pakistan. And in Pakistan, there are certain other factors that add to the effect of neurological disorders, and one of these factors is rate of consanguinity. Consanguinity means the marriages between close relatives. We usually call it cousin marriage. So there is almost over 60% rate of cousin marriages, and it is established fact that the consanguineous marriages or cousin marriages are the major cause of inherited disabilities, including neurological disorders. And here in this diagrammatic representation, you can see it is actually color coded. You can see the colors correctly. Pakistan is among the countries with highest rates of consanguinity. So due to the highest rates of consanguinity and due to the established fact that cousin marriages are one of the major causes of inherited disabilities, I would say that there is a need to incorporate routine genetic testing in healthcare systems for diagnosis and management of neurological disorders. And I will support my argument with the help of three selected families with neurological disorders that we have genetically analyzed in our lab. The first family was presented with severe intellectual disability with progressive decline in ambulation ability to walk. So these are the major symptoms that were presented in the family, and here is the family pedigree. The double line, most of the people should know, represents the cousin marriage, and the individuals marked with the star were available for genetic analysis. On the basis of genetic analysis, we have identified a missense mutation in AIMP1 gene, and that was taking the amino acid glycine to arginine at the position 299, and we have assessed the effect of this mutation using different in silico tools. I am not going to discuss or mention those tools in detail here, but I would like to tell you that different in silico tools predicted this variant to be pathogenic, and it is the disease causing variant segregating in the family. So this was in vivo study performed by some other group that supports the pathogenicity of our variant. The second family was with Magellan-Spelling leukoencephalopathy with subcortical cyst. We can abbreviate it as MLC1. That was a large family with multiple loops with affected people. The major clinical features were muscle spasticity, macrocephaly, extended forehead, joints, contractures, and stiffness. And these were the individuals that were available for genetic analysis. The individuals marked with orange, they were Sanger sequence, and the other one were exon sequence. However, on the basis of genetic analysis, we were able to identify a pathogenic variant in MLC1 gene, and which was changing the amino acid tyrosine at 278 position 16, and different in silico tools also predicted this variant to be pathogenic. This gene is actually responsible for the maintenance of iron and water balance in nerve cells, and a homozygous mutation in this gene would result in absence or destruction of iron and water balance and ultimately lead to the symptoms of this disease, which are brain edema, subcortical fluxes, and myelin and astrocyte deprivation, or you can say the accumulation of fluids inside the cells due to disruption in iron and water balance. The third family was affected with giant axonal neuropathy 1. We suffered this family from KP, district Swat, and these were the major clinical features which were present in this family: progressive loss of ambulation ability. At progressed stage, the individuals were bedridden, the pelvis was deformed, reduced sensitivity, neurally, and this is the family pedigree. Individuals with red stage were available for genetic analysis, and on the basis of genetic analysis, we found a pathogenic variant in G8 gene, exon 9 of this gene. And this variant, we are quoting this variant for the first time from the Pakistani population. In literature, we can find one report of this variant already. This was reported from China in compound heterozygous form. So this is the first homozygous report and the first report from Pakistani population. So in these three families, we not only increase, or we can say we broaden the genetic spectrum of these disorders, but the major conclusion that we can take from these findings are the association of neurological disorders in consanguineous families with pathogenic gene variants underscores the routine genetic testing. The integration of genetic screening into healthcare system can definitely help in better diagnosis and management strategies. So therefore, this study advocates the implementing widespread genetic screening initiatives to improve the patient outcomes and reduce the disease burden in consanguineous population like of Pakistan. Thank you so much.